Welcome again to BioBits and Bytes.
Great to be here.
Today we're diving deep into, well, a really fascinating intersection, immunology and cancer therapy. How our immune system can maybe be engineered, more precisely, to target and beat cancer.
Yeah, it's a hot area. And the research you shared highlights a potential breakthrough, a new platform.
Right, this thing called TRACeR-I. It sounds like it aims to sharpen our immune defenses by cleverly using features of MHC class I antigens.
Exactly. MHC class I. We should probably start there.
Good idea. So we're going to unpack what these MHC class I molecules are, why they're so vital, against cancer viruses, and then how TRACeR-I fits in.
And also how cancer tries to hide by messing with MHCI and how this tech just might get around that.
Okay, so our mission today, really get the core science of TRACeR-I, what the research suggests it could do, and what's next.
Sounds good. So MHC class I antigens, where to begin? Well, MHC is major histocompatibility complex.
Also called HLA in humans, right? Human leukocyte antigens.
That's the one, HLA. And picture this. Almost every cell in your body, the ones with a nucleus anyway, has these MHC class I molecules sitting on its surface.
Okay, so they're everywhere, like little flags.
Kind of. Think of them more like tiny display windows. They're constantly showing little snippets, fragments of proteins from inside the cell.
Displaying what's going on inside.
Precisely. Their main job is presenting these protein bits to the immune system, specifically to CD8 plus T cells.
The killer T cells.
Those are the ones. So if a cell gets infected, say by a virus, or if it turns cancerous, it starts making abnormal proteins, right?
Proteins that shouldn't be there.
Exactly. And MHC class I grabs fragments of these abnormal proteins and puts them in that display window on the cell surface.
Ah, so those are the foreign fragments, the antigens, the red flags we talked about.
You got it. Peptides, antigens, those are the signals that something's wrong inside.
And when those red flags go up on the cellular display window?
That's when the killer T cells, the CD8 plus T cells, notice they're patrolling, checking these windows constantly.
Okay. And if a T cell recognizes one of these foreign peptides stuck to an MHC class I molecule, it basically says, okay, this cell's a threat.
And then zap.
Pretty much.
Wow.
It triggers the T cell to kill that infected or cancerous cell. It's a really critical surveillance system.
It does sound very elegant, very precise. Now, the sources mentioned HLA genes are highly polymorphic. What does that mean?
Polymorphism, yeah. That's key. It just means there are tons of different versions, alleles of these HLA genes. The main ones are HLA-A, B, and BC.
So everyone's display windows are slightly different.
Kind of.
Yeah.
Each HLA version has slightly different preferences for which peptide fragments it can actually bind and display effectively. Think of it like different shaped slots. Only certain shaped keys or peptides fit well.
Okay, and this diversity is good for us overall.
Hugely important for us as a species. It makes it much harder for one single virus or bacteria to just wipe everyone out because our immune systems recognize threats differently.
But it makes designing therapies tricky. Exactly. Targeting something that works for everyone or even most people when their HLA types are so varied, that's been a massive challenge, and that's where TRACeR-I comes in.
Okay, perfect lead-in. Let's get into TRACeR-I. What is it, and how is it aimed to sort of bypass that diversity problem?
Right, so the researchers behind TRACeR-I had this really smart insight. They realized that even though there are loads of different HLA alleles presenting different peptides, the actual basic structure, the 3D shape of the MHC class I molecule itself, the part that holds the peptide, it's actually pretty consistent. There's a limited number of ways it folds up.
Ah, so they're focusing on the holder, not just the thing being held.
You've got it. TRACeR-I is basically a platform, a system for creating molecules, I call them binders, that interact with the MHCI molecule in a way that covers the whole peptide that's being presented.
Okay, how does that help?
Well, the key idea is this PMHCI binder scaffold. Think of it like a foundational structure, like a chassis, that has a surface you can then customize.
Customize for what?
To specifically bind to different disease-related peptides when they're being presented by MHCI. So instead of starting from scratch to target every new peptide on every different HLA type.
Use this common scaffold and just tweak the binding part.
Exactly. The research suggests this could be way faster, potentially more cost-effective for developing these targeted therapies. It aims for a more universal approach.
That does sound efficient.
Yeah.
So how did they test if this actually works? Did it, bind specifically?
Good question. They tested it against 3 quite different peptide MHCI targets. These peptides came from, esophageal cancer, the Epstein-Barr virus, which is linked to some cancers, and SARS-CoV-2, the COVID virus.
A diverse set. And different HLA types, too.
Yep, they made sure the peptides were presented on 2 different common HLA allotypes, HLA-A02 and B08, to really see if it was versatile.
Okay, and the results. Did TRACeR bind?
It did. The binding part, the tracer itself, specifically bound to all 3 of those targets, and it worked across both of the HLA types they tested.
Wow, okay. That's pretty convincing.
It was definitely encouraging. It suggests TRACeR-I could be, a generalized platform. And another crucial thing, safety.
Right. You don't want the binder itself causing problems.
Exactly. Even though these tracers were originally derived from bacteria, they didn't seem to trigger a strong immune response when tested in mice. That's a really important hurdle to clear early on.
Definitely good news. So broadly applicable, specific binding, may be safer. What about making it do something? One source mentioned a BiTE, a bispecific T-cell engager.
Right, the BiTE. Okay, so a BiTE molecule is a type of immunotherapy. Think of it like a microscopic matchmaker or maybe a bridge.
A bridge between what?
Between a T-cell and a cancer cell. One end of the BiTE is engineered to grab onto a specific target on the cancer cell. The other end grabs onto a T-cell.
And it physically pulls them together?
Pretty much. It brings them into really close contact. The idea is to force the T-cell to recognize and attack the cancer cell it's now stuck to.
Okay, I get it. So how did they use TRACeR-I for this?
They took their TRACeR platform, the part that recognizes the peptide MHC complex on the cancer cell, and they built it into a humanized antibody fragment in this BiTE format.
So the TRACeR part is doing the cancer cell targeting for the BiTE.
Exactly. And when they tested this TRACeR-based BiTE, they used actual cancer cells taken from patients, and they saw really effective T-cell activation. The patient's own T-cells got switched on and started killing the cancer cells.
That's fantastic.
And it worked at really low concentrations, nanomolar levels, which suggests it could be quite potent.
Potent and specific.
Mm-hmm.
But how does TRACeR manage that specific recognition? You mentioned the structure. Did they look closer?
They did. They used the technique called X-ray crystallography. Basically, it lets you see the detailed 3D structure of molecules almost down to the atoms.
So I've got a picture of TRACeR grabbing the MHC peptide complex.
Essentially, yes. And it showed something really interesting. TRACeR doesn't just grab the peptide or just the MHC. It binds along the entire length of the peptide MHC-I complex. It fits like a glove over the whole thing.
The whole structure.
The whole thing. And they found this set of 8 amino acids within the TRACeR binder that are pretty much constant. They don't change much, even when targeting different HLA types.
An invariant binding mode, the paper called it.
That's the term, and it's really key because it means the binder doesn't need a totally different strategy for recognizing each target. It has this core way of latching on.
Like a master key system almost.
A bit like that. But here's the clever part. While those 8 residues provide the main anchor, slight variations within those core residues allow TRACeR to fine-tune its grip.
Fine-tune how?
To specifically recognize a wide variety of different epitopes, those specific peptide bits presented by all sorts of different HLA allotypes linked to different diseases.
So it's got this core grip, but subtle changes give it specificity for lots of targets. Point substitution resolution.
Exactly. It suggests they could potentially design tracers for a huge range of disease antigens without losing that specific targeting ability. It seems really adaptable.
Incredibly versatile. Yeah. Now, they also looked at another type of immunotherapy, right?
RRT cells.
Yes, RT cells. Another major player in immunotherapy.
Can you give us the quick rundown on CAR-T?
Sure. You take a patient's own T cells out of their body. Then, in the lab, you genetically engineer them.
Engineer them how?
You give them a new artificial receptor on their surface, a chimeric antigen receptor, or CAR. This CAR is designed to recognize a specific protein found on the surface of cancer cells.
So you're basically programming the T cells to hunt a specific target.
Precisely. Then you grow millions of these engineered cells and infuse them back into the patient. They circulate, find the cancer cells with that target protein, and attack.
Okay, so where does TRACeR fit in with CAR-T?
Well, the researchers created a smaller, single-molecule version of TRACeR, a monomeric form. They tested if this could be used with CAR T cells.
As the targeting part for the CAR.
Sort of as an adapter, almost.
Yeah.
They found this monomeric TRACeR binds really well, high affinity to the CARs on the engineered T cells.
Okay, and then what happens? That binding the TRACeR connecting the CAR T cell to the target MHC peptide complex on the cancer cell triggers the CAR T cell to activate and kill that cancer cell.
So it could potentially use TRACeR's ability to see those internal antigens presented by MHC rather than just surface proteins which CAR-T usually targets.
That's a really interesting potential application, yes. It could expand the range of targets for CAR-T or maybe enhance their ability to recognize cells even if the usual CAR target isn't present at high levels.
Okay, this all sounds very promising.
Hmm.
But cancer's tricky. The sources really emphasize how cancer cells evade the immune system. We mentioned MHCI downregulation, taking down those kick-me signs.
Exactly. It's a huge problem. Tumors are masters of disguise and evasion. Reducing MHC class I presentation is one of their main tricks to avoid those killer T cells.
How do they do it? Is it just one mechanism?
Oh no, they have a whole toolkit. It can be through actual genetic defects mutations or outright loss of the HLA genes themselves, or other genes needed to process and load the peptides, like Me2M or TP. Sometimes whole chunks of chromosome 6, where HLA genes live, get lost.
So permanent damage to the system.
That's one way, but they also use sneakier, sometimes reversible methods. Epigenetic silencing, basically switching off the MHCI genes without actually damaging the DNA sequence.
Like hiding the instruction manual instead of shredding it.
Good analogy. They can also mess with the proteins that normally turn on MHCI genes or use tiny bits of RNA, microRNAs, to block the MHCI messages from being turned into proteins. Or sometimes they just stop making the specific tumor antigen that the immune system was looking for.
Wow. Multiple lines of defense against being detected.
It really is. And this loss or reduction of MHCI, it's bad news for patients. It's linked to worse outcomes and makes many immunotherapies, like checkpoint inhibitors, less likely to work.
Because the T cells just can't see the target anymore.
Exactly. But the fact that some mechanisms, those soft lesions like epigenetic silencing, might be reversible, that offers a glimmer of hope. Maybe we can force the cancer cells to show their flags again.
Which brings us back again to TRACeR-I. Given this big challenge of MHCI hiding, how might TRACeR-I offer a way forward?
Well, a few ways, potentially. First, TRACeR-I binds the peptide and the MHC molecule. So even if the number of MHCI molecules on the surface is lower, TRACeR might still be sensitive enough to find the ones that are there and still effectively target the cell.
So it might work even with fewer flags flying.
That's the hope. The BITE results, where it killed patient cancer cells, suggest it can overcome at least some level of downregulation. Second, its adaptability.
Because they can generate new binders relatively quickly.
Right. If the tumor changes the peptides it's presenting to try and escape, maybe we can develop new tracers fast enough to keep up, targeting the new peptide MHC complexes.
Staying one step ahead, maybe.
Or at least keeping pace. And third, that monomeric version for CAR-T. Combining TRACER's specific MHC peptide recognition with the killing power of CAR-T could be a potent combo, potentially working even if MHCI levels are partially reduced. It adds another layer to the attack.
It really sounds like it's trying to tackle the problem from multiple angles. Now, the sources also mentioned predicting MHCI binding, a tool called HL-Apollo. How does prediction fit in?
Prediction is hugely important. To design any therapy that targets these peptide MHC complexes, whether a vaccine or something like TRACeR-I, you first need to know which peptides the cancer cell is actually presenting.
Which flags it's actually putting in the window.
Exactly. And predicting that is complex because of all the different HLA types and the vast number of potential peptides inside a cell.
So HL-Apollo is trying to improve those predictions.
Yes. It's described as a newer transformer-based model using AI techniques. It looks not just at the peptide sequence and the HLA sequence, but also pulls in information about the protein the peptide came from.
Giving it more context.
Seems like it. And the research suggests it's more accurate than older methods and better at handling real-world samples that might have multiple HLA types mixed together. It's even trying to tackle predictions for rarer HLA types.
That sounds crucial for making therapies more personalized or broadly applicable.
Absolutely. So while TRACeR-I is the thing that does the binding, tools like HL-Apollo are essential for telling us what to target. They help identify the best peptide MHC addresses for TRACeR-I to go after.
So the predictors find the target, and TRACeR-I provides the weapon system.
That's a good way to put it. It's a synergistic relationship.
Yeah.
Better prediction leads to better target selection for platforms like TRACeR-I.
Okay. This is all very exciting. But the researchers themselves said more work is needed, right? What are the next big hurdles for TRACeR-I?
Definitely. Early days still. Key things they mention. Optimizing the actual cell-killing efficacy. Making sure it's potent enough in various settings.
Making sure it really gets the job done.
Right. And safety, specifically immunogenicity in humans. The mouse data was good, but humans are different. We need careful preclinical work and eventually clinical trials to ensure it doesn't cause unwanted immune reactions.
Always a major checkpoint for any new therapy.
Absolutely. And finally, looking at long-term performance in vivo in living systems over time. Does it keep working? Is it durable? Those are critical questions before it could become a real therapy.
So a lot of work still ahead, but the foundation seems really interesting. This deep dive has been fascinating, seeing how understanding this basic MHC system is leading to these innovative therapeutic ideas like TRACeR-I.
It really highlights that connection between fundamental biology and clinical application.
And the potential for TRACeR-I to be, maybe more adaptable, maybe easier to develop, combined with better prediction tools. It really does paint a promising picture for the future of targeting these intracellular flags.
It does. It's tackling some long-standing challenges in immunotherapy head-on.
Well, for you, our listener, we really hope this exploration has sparked your curiosity about where cancer research is heading and the sheer ingenuity involved in harnessing our own immune system.
It's a constantly evolving field, always something new to learn.
Indeed. Thank you so much for joining us on this deep dive here on BioBits and Bytes.
Thanks for having me.
We look forward to our next exploration together.